look at properly. Transpiration and water transport. It seems a bit random that I've just sort of gone on to plants. Basically, what I'm doing is sort of following on from where we got to with lungs and sort of that gas exchange system. So we'll just have a look at sort of exchange systems in plants. So sort of a little bit of a sidetrack away from what we have been doing with sort of human biology. So we're going to look at process of transpiration and water transport, understand what is meant by water potential, think about cohesion and tension theory, explore the different factors affecting the rates of transpiration, learn about how plants conserve water, and understand what is meant by root pressure. So transpiration is the loss of water from the leaves of a plant, and most of this occurs from the underside of a leaf where there are many stomata. Sorry, I just need to get this for 1 second. So transpiration is that loss of water, like I was saying, and it often happens on the epidermis, which is where we've got lots of stomata, and that's on the underside of the leaf of a plant. And actually, most plants control their water intake because they're able to open and close their stomata. So as water levels change, they have some special cells around the stomata, and these are called guard cells, and these are able to open and close the stomata. And this occurs either passively by osmosis or by active transports of solutes, and that's when the water levels changes, and so the guard cells are able to open and close them. So transpiration rates also vary naturally in response to environmental factors, and so that's obviously down to things like temperature and humidity. So we talk about water potential a lot when we're looking at transpiration. So water tends to move, as we've said before, with osmosis from areas of high water concentration to areas of low water concentration, and that's osmosis. So water also tends to move from areas of high hydrostatic pressure to areas of low hydrostatic pressure. And it's also affected by gravity and electrostatic forces, such as those that cause surface tension. So collectively, the term for the tendency of the water to move due to any of these effects is something called water potential. So water is a polar molecule, and that means that its positive and negative charges are not evenly distributed. The oxygen atom has a slightly negative charge, while the two hydrogen atoms are slightly positive. And so we've talked about water cohesion before. So again, this is just like a little recap from when we did that whole topic on water. So what this means is that in the xylem, water molecules spontaneously arrange themselves so that the positive and negatively charged poles lie next to each other. And because of this causes the molecules to cohere or stick to each other, so that if some leave a plant by transpiration, others are pulled up behind them. And this is that tension theory. So this is how they pull them up. So we have this constant stream of water through the plant. So how might we investigate rates of transpiration? So you've probably done this before at GCSE. You can use something called a potometer. And a potometer is a device that measures the water uptake of a plant. And this is an image of a potometer. And so what we would often do is we often want to look at the mass as well. So we want to look at the varying mass of the plant because of water uptake. So to compare transpiration rates in different settings, we want to look at the advantages and disadvantages of using the same cuttings and several different cuttings. So if we wanted to compare transpiration rates because of environmental factors, we only want to use one cutting. And that's because different cuttings will have different surface areas. So if you have a different surface area, we know it will affect the total transpiration rate. So using one cutting keeps the surface area of transpiration constant, and less equipment is needed, but it will take a lot longer to collect the necessary data. So how could we prepare the cutting? So what we want to do with the cutting is it should be prepared by cutting the stem with a sharp knife, but specifically underwater. And that's really, really important to stop it from drying out. And that water potential. So what should be done next? So what we do is we seal the newly cut shoot into the potometer, keeping it underwater as we do so. Once we've done that, we can lift it out of the water. So the apparatus now is free of unwanted air bubbles, and we clamp it in place and one bubble is introduced to the capillary tube, and this is really important, this one bubble. And this one bubble is what we use to do all of our measurements. And what happens is, as the cutting takes up water, that bubble moves along the tube. And this is then calibrated to show the distance moved, and in turn, that represents the volume lost. So what readings should we actually take to find out the rate of transpiration in each different set of conditions? So to measure the rate of transpiration in each different set of conditions, we need to take readings every 3 minutes for 30 minutes. So we should have at least 10 recordings. Now here's some example results. So experiment 1. As time passed, we're going to put our recordings on, and this is a distance that our bubble has moved along the capillary tube. Speed that up so we can get to it a bit quicker. And as we can see, we've got an increase in the direction of it moving. It's moved more distance each time. So that was our control. Now we're looking at it with light. So does light affect the transpiration rate? So we've put the light on, and we're going to plot it again. We're going to plot it on the same graph so we can compare them quite easily. And we can see that that bubble is moving a lot further and a lot quicker than when it was just in the control because of the increase in light. So we can see we've got a higher rate of the bubble moving, so high rate of transpiration. And then finally, does air movement affect transpiration? I say finally, it might be another one after this. So we've got air movement going, and then we can mark these on. And actually, these are fairly similar. There's a slight difference, but they're not too dissimilar to each other. Oh, and humidity. So if you look at humidity, speed this up a bit, and we do have a difference due to humidity, but that is lower than our control. So the rate of transpiration was most strongly affected by, as we can see from this diagram, light. This increased the rate of transpiration by causing some stomata to open. So how does water get from the plant's roots to its leaves? So we're going to look at how this happens. So plants lose water from their leaves by that process called transpiration. And this draws water up the stem in a constant stream. So what happens is, as we know, our root is lined with layers of cells called the epidermis. And many of these cells feature a long outgrowth, which we call the root hair. The root hair is great because it increases surface area for osmosis to occur in. So what happens is the water now must move through the main bulk of the root, the cortex, to reach the xylem. It may travel via the symplast pathway, moving between cells via pores in cell walls called plasmodesmata. Or it may travel through the cortex via the apoplast pathway, moving by osmosis through the cellular matrix of the cell walls until it reaches the endodermis. The endodermis separates the root cortex from the stele, which contains the xylem. Here, the apoplast pathway is blocked by a coating of suberin on the cell walls known as Casparian strip. The Casparian strip gives the plant more control over what enters the xylem, as water must go through the cell membrane of the endodermal cells, which is selectively permeable. Water is now able to enter the xylem, and once it's able to enter the xylem, it begins to rise up the root and towards the stem. And because it's going against gravity, it's drawn up using cohesion of water molecules to each other. And as water leaves the plant by transpiration, the cohesion of water pulls the water up and through and allows this transport stream. When the water reaches a leaf, it moves out of the xylem and into the surrounding cells by osmosis, and then water vapor enters the air spaces in the spongy mesophyll. Water vapor finally diffuses out of the plant via the stomata, and this is the process known as transpiration. The stomata open and close in response to factors such as incident light and availability of water. So we're just going to look at an example of how different plants might be adapted to dry conditions. So we're going to look at a plant called prickly pear, so Opuntia, and it belongs to the cactus family, and they are native in the Americas, and they are xerophytes, which means they're adapted for environments where there's little water available, so it's an extremophile. So one of the ways they're adapted to conserve water is they have spines, and the spines on a cactus protect it from predation, which it is susceptible to because it is a good source of water. And they also reflect some light to lower the internal temperature of the plant. And they may even be able to trap a layer of air at the plant's surface to reduce evaporation. Then we've got the epidermis. So the epidermis of a prickly pear is covered in a waxy layer known as the cuticle. And this is thicker than that of the average plant, and that's to help reduce evaporation through the tissue surface. Then we've got the stomatum. Stomata and cacti are really, really clever. They're actually located around the stem, and we have a lot less of them on Opuntia and any cactus compared to normal plants. And most plants, they open all the time, open and close. It doesn't have to be day or night. But on cacti, they open at night rather than during the day, and this is to reduce water loss. The carbon dioxide is absorbed then and then fixed for using during the day when the plant is photosynthesizing and the stomata are closed. And finally, the stem. The Opuntia display only very small temporary leaves, and the majority of photosynthesis actually occurs in the stem, and that forms a thick, fleshy pads, and they are adapted for efficient water storage. And channels of pale mucilage cells hold additional water. So the other thing we have to look at is something called root pressure. So water is usually drawn up a plant by the tension resulting from transpiration and cohesion between water molecules. In some situations, such as 100% humidity, a plant is unable to transpire. Instead, water can be transported by positive pressure from below, and this is known as root pressure. So solutes are actively transported into the roots of the plant, causing water to enter by osmosis, and this increases the hydrostatic pressure in the root, forcing water up the stem. So let's end that. And that's our little sidetrack to transpiration. And now I just want to go back to there, and we're going to have a look at digestion and absorption. No, I don't want to do that. No. Let's just go back to the AS biology. Organisms, lifestyle disease. We've done lifestyle disease, haven't we? Osteopathogens, lung disease. Yeah. We're just going to quickly have a quick look at blood vessels. So we've done the circulatory systems. We're just going to have a quick look at blood vessels. So we're going back to humans now. So a bit of a sidetrack and then back again. So blood vessels. Going to be a really quick overview of blood vessels. Nothing really new to what you've learned at GCSE. So quick overview now. Okay, so structure of blood vessels. And we're going to look at the structure of blood vessels and how it's closely related to their function. So arteries. Arteries, unless we're talking about the pulmonary artery, they always carry blood away from the heart. Well, no, they always carry blood away from the heart. Ignore me about pulmonary arteries. They always carry oxygenated blood, unless it's a pulmonary artery and it carries deoxygenated blood. And blood is pumped out of the heart at a really, really high pressure. So arteries are structured to withstand and maintain this really high pressure. So to do this, they have the lumen. Lumen is the area where all the blood flows through it. So it's the empty cavity. And the lumen of arteries is actually really narrow. That's to help maintain that really, really high blood pressure. If it was too wide, then that blood pressure would drop. So they have a really narrow lumen for high blood pressure. The endothelium, this forms the inner layer of an artery. So it's also known as a tunica interna, and it consists mainly of endothelial cells. So we've got that. And we've got our smooth muscle cells and elastic tissue. And this forms the middle and the thickest layer of an artery wall. And it's also known as a tunica media. This enables our arteries to flex and to withstand that and maintain that high blood pressure. If they weren't able to flex, they would snap and they would break because of such high pressure that blood is traveling through our arteries. So they have to have that flexibility to them. Then they have connective tissue, and this makes the outer layer of the artery wall, and it's also known as a tunica externa. And this tissue consists mainly of tough collagen and fibers, and these provide strength and durability. So we're now going to look at the arteriole. So we have our arteries, and they're carrying all our blood away from the heart. And then we have an arteriole, and these branch off from our arteries. They're actually very, very similar in structure to our arteries, but they have less elastic tissue and they have more smooth muscle. And they enable them, this enables them to constrict, and they actually reduce blood flow through the capillaries, because we don't want the blood flowing through the capillaries at the same pressure as it's flowing through arteries, otherwise our capillaries would end up being damaged. So arterioles take the blood from the arteries to the capillaries. So then we have capillaries. So capillaries branch off from our arterioles, and they are our smallest of all blood vessels, and they form our capillary beds, and they function to bring the blood as close to cells as possible to enable that efficient exchange of substances. So the lumen of capillaries is super, super narrow. It's about 7 micrometers in diameter. So essentially that's one red blood cell can pass through at a time. So this really slows the blood flow down, and this allows substances to diffuse in and out. Otherwise, it'd be flowing so fast that the substances wouldn't be able to diffuse in and out effectively. Also has an endothelium. It's really, really thin. It's only 1 cell thick, and that's to allow that efficient diffusion. That's one of them things, those questions that you always get. How is gas exchange? How the lungs adapt to gas exchange? How the capillaries adapt to gas exchange? Or the adapted for gas exchange, because their endothelium is only 1 cell thick. Basement membrane, and the basement membrane anchors our capillaries in position within tissue, so they're not all floating around, they're in a set place. That takes us onto our venule, and these drain blood from the capillary beds and they feed it back into the veins. Again, they contain smooth muscle, and they can constrict like arterioles, and this is to control the blood flow through the capillaries. And finally, we have the vein. So vein transports blood back to the heart. Generally, it's deoxygenated blood unless it's the pulmonary vein. The blood pressure in veins is a lot lower than in arteries, and so this has a big impact on their structure compared to arteries. So the lumen of veins is actually really large in comparison to the lumen of arteries, and that's because they don't need to maintain that high blood pressure. They can have a more open, spacious lumen. The endothelium is actually really similar to that of the arteries. The smooth muscle elastic tissue layer of veins is actually a lot thinner, and that's because they don't need to withstand or maintain that high blood pressure that we find in arteries. So they're not going to basically get battered about by that high pressured blood as much, so they don't need as much elasticity and flexibility. And then finally, their connective tissue is thinner in arteries because, again, they don't need to withstand that high blood pressure. And they have one extra thing that arteries don't have, and that is a valve. So veins contain valves, and that's to prevent backflow of blood away from the heart. It also helps to prevent blood from pooling in the lower parts of the body due to gravity. So without them, we could get things like blood clots forming as well. So identifying blood vessels. So which of the 2 larger vessels is an artery and which is a vein? So over here, we've got a vein, and over here, we've got an artery. We can see that because we've got the lumens here. The lumen of the vein is much larger than the lumen of the artery. Smooth muscle is a lot larger in the artery than the vein, and the connective tissue is different as well. So how does blood actually flow through our veins? Let's have a quick look at that. So blood is able to go through our veins through contraction. So what happens is our skeletal muscles are able to contract. And when they contract, blood beyond the site of contraction is actually forced towards the heart. So if we watch this again, so if it's below that bit's forced towards the heart, and the valves behind the site of contraction prevent blood from being forced away from the heart. So again, they close and they stop them from being forced away, and it pushes it back up. Now, when they relax, as skeletal muscles relax, blood pushes back against the valves, and that causes them to close, and this again prevents the blood from being forced away from the heart again. So something you've probably heard about or heard of is something called a varicose vein. Now, a varicose vein occurs if the vein wall becomes weakened, and if this happens, valves might not actually be able to close properly. And if this happens, a backflow of blood can occur, and this causes the vein to become enlarged and bumpy, and we call this varicose. This usually happens in superficial veins, so that means veins close to the skin, so that often happens in the lower legs, as opposed to deep veins, which we find underneath muscle. Varicose veins can be surgically removed without affecting blood flow, as most blood is returned to the heart by deep veins, not those superficial veins. So how does our body maintain this high blood pressure? So blood pressure is the main force that drives blood from the heart and around the body. So during systole, heart contraction, blood is pumped through the aorta and other arteries at high pressure. The elastic fibers of arteries enable them to expand and allow blood through. And during diastole, heart relaxation, the blood pressure in the arteries drops. The elastic recoil of the artery walls help force the blood on. So as blood moves through smaller arterioles into capillaries and then into venules and veins, its velocity and pressure drops continuously. So done blood vessels. Let's have a quick overview of blood tissues and blood tissue fluid. So blood is a specialized transport medium that is also considered a special type of connective tissue. Most adults have between 4 to 6 litres of blood in their body. Blood has a huge range of functions. It can be used for transport, defense, thermoregulation, and maintaining pH of body fluids. So what is in blood? Again, just a quick recap from GCSE. So 45% of our blood is made up of cells. So that can be red blood cells, also known as erythrocytes. So these transport oxygen, and we have 4 to 6 million per millimeter cubed. We have leukocytes, white blood cells, and these are involved in immunity, and we have 4,000 to 11,000 per millimeter cubed. And then finally, thrombocytes, which are platelets, and these are involved in blood clotting. We have 200,000 to 500,000 per millimeter cubed. 55% of our blood is made up of plasma. Around 92% of that is water, and the rest of that is composed of proteins such as albumins, fibrinogen, and antibodies, ions such as sodium ions, potassium, calcium, and chlorine, nutrients such as glucose, amino acids, and fatty acids, waste products such as urea, hormones, for example, insulin and estrogen, and then gases, oxygen and carbon dioxide. So, erythrocytes, red blood cells. What are some specialist parts of them? Again, nothing should be super new about this. So they have this really special shape to them. We call it a flattened biconcave shape. What this shape does is it allows them to have a really, really large surface area to volume ratio. And that helps in gas exchange. You'll start to notice that any sort of substance exchange in biology, an answer is it has this adaptation. It has a specialized feature to allow it to have efficient gas exchange, large surface area, efficient gas exchange. They have a large amount of hemoglobin, and that allows them to transport oxygen. They have absolutely no nucleus or organelles, and that's to maximize space for hemoglobin so more oxygen can be transported. And their diameter is between 6 to 8 micrometers, and that is larger than capillary diameter. So this slows blood flow to enable diffusion of oxygen. So we're going to have a quick look at blood clotting now. So coagulation is when blood sits together and it forms a clot. So we're going to look at how that forms. So how does a blood clot form? So in our body, we have circulating anticoagulants such as heparin, and these normally prevent our blood from clotting. However, when something such as the wall of blood vessel is damaged, the collagen fibers become exposed to the blood. When this happens, those platelets, the thrombocytes, they come along and they stick to the exposed collagen fibers, and they do this to form a plug to stop the bleeding. The platelets become activated and they release a clotting factor called thromboplastin. And then through a complex cascade, thromboplastins convert something called prothrombin, an inactive plasma protein, into thrombin, which is an active plasma protein. This process requires calcium and vitamin K. So then we get the platelet plug forming, and the thrombin catalyzes the conversion of the soluble fibrinogen into insoluble fibrin. Fibrin forms a fibrous mesh that then traps red blood cells and strengthens that platelet plug, and this forms a clot. So the blood clot then is eventually reabsorbed by another complex process called fibrinolysis. And the main enzyme that controls this process is called plasmin. And that is our blood clot being reabsorbed, because we don't want that to stay there forever. So summary, we get a damaged blood vessel, and that exposes some collagen. That exposed collagen has platelets that come and attach to it. So inactive platelets attach to exposed collagen, and when they attach to exposed collagen, they become activated. When they are activated, that means they give off thromboplastins, and with thromboplastins and vitamin K and calcium, they react and make prothrombin. Prothrombin forms thrombin, and thrombin allows fibrinogen to become fibrin, which adds to that platelet plug and helps form that blood clot. So, plasma proteins and blood pressure. So about 8% of blood plasma consists of plasma proteins, of which half may be albumins. These are a group of small proteins involved in the transport of other substances, so for example, fatty acids and hormones, and they help regulate the osmotic pressure of blood. The balance between the hydrostatic pressure of blood and the osmotic pressure of blood is important in the formation of tissue fluid. So how is tissue fluid formed? Oops. All cells are bathed in tissue fluid, and that enables substances to diffuse in and out. So blood at the arterial end of the capillary is at a relatively high pressure, and this forces water and other small molecules out, and this is through the capillary wall by a process called ultrafiltration. This is tissue fluid, and it moves into the spaces between cells. It's similar to blood, but it doesn't contain plasma proteins such as albumin, as they are too large to pass through the capillary wall, so we call it tissue fluid. So then the plasma proteins that remain in the blood exert an osmotic effect, which draws some water back into the capillary, but not enough to counteract the loss due to blood pressure. At the venous end of the capillary, the blood is under low pressure, which means the osmotic pressure exerted by plasma proteins is greater than the blood pressure. The net flow of water is therefore into the capillary from the spaces between cells. So not all tissue fluid returns to the capillaries. The excess tissue fluid drains into our lymphatic system and it forms lymphs. Lymph is a colourless pale yellow fluid, and it's similar to tissue fluid, but it contains more lipids. That's the only difference. And the lymphatic system drains into the circulatory system near the vena cava via the thoracic duct. The lymphatic system is a second circulatory system, and it is a major part of the immune system because it contains the lymphatic capillaries and vein-like lymph vessels, and these contain valves. Lymph nodes, and these are sac-like organs, and these trap pathogens and foreign substances, and they contain large numbers of white blood cells. And lymphatic tissue, and this is in the spleen, the thymus, and the tonsils, and these also contain large amounts of white blood cells and are involved in their development. So, I think we might have done this actually. No. So oxygen transport around the body. Again, we'll have done a little bit of this, but we'll super recap basically. So oxygen, to be transported around the body, we need this molecule called hemoglobin. hemoglobin is a protein and it actually makes up 95% of the dry mass of a red blood cell. It is the means of transporting oxygen around the body. And hemoglobin is made up of 4 polypeptide chains, and each one is bound to a heme group. Each heme group can combine with one oxygen molecule so that one molecule of hemoglobin can combine with a maximum of 4 oxygen molecules, and this forms something called oxyhemoglobin. Oxygen binds to hemoglobin when oxygen is at a high concentration and dissociates from hemoglobin when oxygen is at a low concentration. The concentration of a gas in a mixture of gases can be quantified in terms of its partial pressure. This is the amount of pressure exerted by the gas relative to the total pressure exerted by all the gases in the mixture. So partial pressure is measured in kilopascals, something that you will have seen in chemistry before. So the oxygen dissociation curve is a graph that shows the percentage saturation of hemoglobin at various partial pressures of oxygen. So at low partial pressure, the ratio of oxyhemoglobin to hemoglobin is correspondingly low. Most hemoglobin molecules will only take up one oxygen molecule or none at all. So once a hemoglobin molecule has picked up one oxygen molecule, its oxygen affinity increases. Therefore, there's a small increase in oxygen availability, thus it leads to a large increase in saturation and the graph gets steeper. Once a high saturation is reached, some molecules will be unable to take on any more oxygen. So further rises in partial pressure thus makes less difference to the saturation and the graph begins to level out. In the lungs, the oxygen partial pressure is high, so most hemoglobin becomes saturated with oxygen. The normal range of partial pressure in the lungs is shown by the shading on the graph that we've got here. So this is our normal partial pressure in our lungs. And in metabolizing tissues, the oxygen partial pressure is much lower. So the oxygen saturation of hemoglobin drops dramatically and releasing oxygen into the tissues. The result is the transport of oxygen from the lungs to tissues. So what affects oxygen dissociation? So we have something called the Bohr shift and temperature dependence. So as we have an increase in carbon dioxide in a tissue, such as during exercise, this lowers its pH. This lowers the oxygen affinity over all partial pressures and leads to an increased release of oxygen where it is needed. And we call this the Bohr shift. So again, lowers the oxygen affinity over all partial pressures, and that's the Bohr shift. And a decrease in temperature actually increases hemoglobin's oxygen affinity slightly. So this means that hypothermia reduces the blood's ability to release oxygen into metabolizing tissues. So fetal hemoglobin, so the red blood cells that we in the fetal bloodstream contain a special form of hemoglobin known as fetal hemoglobin. And fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. This helps maximize oxygen uptake from the mother's bloodstream, which has already lost some of its oxygen by the time it reaches the placenta. Myoglobin. So myoglobin is a molecule with similar structure to hemoglobin, but with only one heme group. Myoglobin has a very, very high affinity for oxygen, even at very low partial pressures. This means that oxymyoglobin will only dissociate with oxygen levels when they are very low. This is found in muscle cells where it acts as an oxygen reserve. Okay, we're getting through this actually quite quick today, so I'm going to do one more little bit. Oxygen transport, blood will do organisms, heart, circulation, transport in plants. We'll go back to plants really quickly. And we're going to look at transport systems in plants. So smaller primitive plants such as mosses, they can just get all their nutrients from the environment. They're able to absorb like that, like really small animals. However, large plants don't have a large enough surface area to do this. So like most multicellular animals, they've developed specialized tissues for transporting water and nutrients to cells. Plants that have specialized transport systems are called vascular plants. So common features of vascular plants. So Water and dissolved minerals are transported by the xylem. The root, outgrowths, are called root hairs, and they give the root a large surface area. Water is able to travel up the root's cortex, and that consists of relatively undifferentiated parenchyma cells into the vascular tissue in the stele. Then we have xylem. So xylem allows for water to move through its pores, or sometimes called pits, and in elongated tracheide cells, while vessel elements with perforated end plates join end to end to form continuous vessels, and the sclerenchyma fibers provide support. In the leaf, vascular bundles surrounded by mesophyll tissue, and these contain airspace into which water evaporates. Water vapor then leaves the plant via the stomata, which opens and closes to control water loss. Sugars, such as sucrose, are transported via the phloem. We also have a tuber. So tubers such as potatoes are enlarged roots, and these store sugar and they convert them into starch. The tuber grows as a plant is photosynthesizing, but it will then break down again to provide sucrose when it's needed for growth. And the phloem consists of living cells. So xylem is made up of dead cells, phloem is made up of living cells, and these are known as sieve tubes. Each one is supported by something called a companion cell, as it lacks some organelles. To allow sap to move easily, sclerenchyma fibers again provides support of these, and then we have fruit. So fruit stores sugars by converting them to starch, but unlike tubers, they do not provide energy for the growing plant. They provide energy for the daughter plant and attract the animals that distribute their seeds. So if we have a look at this dicotyledon root here, we've got a phloem, which we can see here, an endodermis and a xylem. So that is our phloem there. So that's quickly our plant overview. Close that. Let's look at translocation of sugar and then we will finish. So translocation, that is a movement of nutrients around plants. It includes the movement of minerals, which can be dissolved in water and transported in the xylem, but usually we refer it to the transport of sugars, amino acids, and other organic molecules that are transported in the phloem. Translocation can occur in either direction of the phloem, and it is bidirectional. It is an active process requiring energy, unlike water transport in the xylem. The most widely accepted explanation of sap movement in plants is the pressure flow hypothesis. According to the theory, sap moves through phloem vessels due to differences in hydrostatic pressure, and this is similar to effect to root pressure. So evidence of this effect includes the excretion of sap or honeydew by an aphid when it taps into a phloem vessel to feed. The sap is forced through the aphid's body, demonstrating that the sap in the phloem is under pressure. So, in our plants, transport of sugars, glucose, that is made in our chloroplasts by photosynthesis. This is then turned into sucrose for transport around the plant, or it can be temporarily stored as starch grains within the stroma matrix for use at a later stage. This sucrose is then moved through the leaf cells and into the phloem by active transport. This is known as active loading, and during this process, it reduces the water potential and it causes water to enter the sieve tubes by osmosis. So this is active loading through the leaf cell, through the companion cell, into the phloem. As water enters the phloem, the hydrostatic pressure then increases. What this does is this forces the phloem sap through the vessel towards the regions of lower pressure. And this is purely physical process. It's known as mass flow. And that's all to do with the xylem and the water going up and the phloem and the sugars going down. So sucrose is actively then unloaded from the phloem where it's needed for growth, or for wherever it's needed for growth, or into a storage organ such as a tuber, where it is converted into starch, and these are then known as sink. So again, it goes through the companion cell and then into whatever cell is needed. So it could be wherever it's needed for growth or into the tuber cell, for example, and that is known as a sink. A sink removes sugar from the phloem and it increases the water potential. So water leaves the sieve tubes by osmosis and it keeps the hydrostatic pressure low. So the STAP continuously flows from source to sink. So we have this continuous flow going from source to sink. So translocation can occur in both directions in a phloem vessel. So for example, in the spring, a tuber can become a source where starch is broken down into sucrose and the growing tips of a plant become a sink. So we are going to leave it there because we've actually covered loads, and that's loads for you to look at. So let me stop for